In this video, I will summarize active transport. Active transport is based on two basic criteria. First one is we go against the concentration gradient. Okay, and to go against concentration gradient, we need energy. And on the basis of source of energy, we classify this active transport into primary active transport or secondary active transport. There is only one major difference, that in case of primary active transport, the energy source has to be direct. The energy source can be light, it can be any nucleotide like adenosine triphosphate, guanosine triphosphate. Okay, it can be light, it can be nucleotide or any other direct source. But if the source of energy is indirect, for example, the gradient established by primary active transport has been used to transport a molecule, we say it is secondary active transport. Another major difference is unipolar can only be used in primary active transport. For secondary active transport, we can only use sympotar or antipotar, whereas unipotar, sympotar and antipotar, they are all relevant in case of primary active transport. There are some rare cases where channels can also be used in case of active transport, whereas in the video of passive transport I described that channels are the passive transporters. But when channels are used as active transporters, they transport along the gradient. Now this goes against the principle of active transport, but still we count this process in active transport because energy is being utilized, and this energy is used to open the gate of this active transporter channel. Let's have a look at the process itself. This is a plasma membrane, this is inside the cell, cytoplasm, this is outside the cell. Okay, we have a molecule whose concentration is higher outside the cell and lower in case of cytoplasm. We have to transport this molecule against the concentration gradient from inside to outside. To do so, we need a transporter protein. Now this transporter protein uses energy directly from a nucleotide. It is using energy from ATP, so this is primary active transport, and this protein is using ATP as energy source, so this protein itself is known as ATPase. Alright, this is hydrolyzing ATP and transporting this molecule against the concentration gradient. This is primary active transport. In case of secondary active transport, the primary active transport is still happening, which is transporting these protons against the concentration gradient, and if it is transporting protons and using ATP as energy source, we say this transporter protein is H positive ATPase. Now this primary transport is establishing a stronger gradient. More and more protons have been accumulated outside the cell. Now these accumulated protons will have potential energy, and these accumulated protons will start moving in along the concentration gradient, and when they move along the concentration gradient, they lose this energy, and this energy can be used by another molecule X to be transported along with this proton against the concentration gradient. This is what secondary active transport is. Now we will see the same process in a more relevant example, the transport of glucose across intestinal epithelial cells. Now have a look at this diagram. We have epithelial cell right here, we have intercellular space on the left, and we have intestinal lumen on the right. Potassium is present in higher concentration inside the cell, same as glucose, whereas sodium is in lower concentration inside the cell. But outside the cell it follows completely different situation, where potassium is low, sodium is high, and glucose is low. Same is happening outside the cell in intestinal lumen as well. Now let's have a look at the basolateral surface. Okay, we have sodium potassium ATPase, which is transporting 3 sodiums outside, which is against the concentration gradient, and it is transporting 2 potassium ions inside the cell, which is also against the concentration gradient. So it is transporting against the concentration gradient, ATP is being utilized. This is primary active transport. Okay, and by doing so it is establishing stronger gradients. Now more sodium will be outside the cell. Similarly here more sodium will be outside the cell, and lesser sodium will be inside the cell. Okay, so this sodium will start moving along the concentration gradient. Okay, and when this sodium will move, it will lose its potential energy, and that lost energy is being used by glucose to go against its concentration gradient. So this is secondary active transport. Now we will have a look at different examples of active transport. We will start with sodium potassium ATPase, the same sodium potassium pump we saw in intestinal epithelial cells. We will basically start with the structure of sodium potassium ATPase. It consists of three subunits: alpha subunit, beta subunit, and gamma subunit. Alpha subunit is multi-pass transmembrane protein, whereas beta and gamma subunits are single pass transmembrane proteins. We will start with alpha subunit. Alpha subunit is 10 pass transmembrane protein, and it is even pass, so both the N and C terminus have to be on same side. In this case both N and C terminus are on cytosolic side. In close proximity to N terminus we have activator domain. Same activator domain can be seen on second and third loop. On the same second and third loop we have nucleotide binding domain. Alright, we have a regulatory domain on the loop of fourth and fifth helix. Beta subunit is single pass transmembrane protein having N terminus on the cytosolic side and C terminus on outside or exoplasmic phase. Okay, on the C terminus the glycosylation can be seen, but in case of gamma subunit there is a certain significance here. The C terminus is inside the cell, N terminus is outside the cell, but near the N terminus we have F, X, Y, D sequence. What is this sequence? F is phenylalanine, X is any amino acid, Y is tyrosine, and D is aspartate. Let's see the working of this sodium potassium ATPase. Okay, you can see in this diagram it starts with binding of 3 sodium ions, and after this the phosphorylation of aspartate will occur. This phosphorylation of aspartate will induce a conformation change. The protein will flip. As you can see here, the 3 sodium ions will be released, the 2 sodium ions will be bound. Here you can see 2 sodium ions have been bound. After binding of 2 sodium ions, this aspartate will dephosphorylate, and by this dephosphorylation a conformational change will occur. The protein will flip again, and there will be release of these 2 potassium ions, as you can see right here, dephosphorylation and release of 2 potassium ion. Now there are certain inhibitors of sodium potassium ATPase or sodium potassium pump, we can say. The major one is digoxin or digoxin. It is extracted from Digitalis purpurea, and it is considered as cardiotonic steroid because it increases the muscle contraction in myocardium. Okay, now which step of the functioning of sodium potassium ATPase this digoxin targets? It targets the dephosphorylation step, and it inhibits the sodium potassium pump, sodium potassium ATPase. Now let's see the 2 different situations. The first situation is where digoxin is not present. Okay, this is the normal functioning of myocardium. And second situation, we have digoxin present. This is the case we will see. But before that, in case of normal cells, we have a well functioning sodium potassium ATPase, which is increasing the sodium ions outside the cell. Now if it is a primary transport, it will create a stronger gradient of sodium ions. These sodium ions will come back, and they come back along the gradient, and the calcium ions will be transported against the gradient in an antipode way. This is antipode, sodium calcium antipoder. Now these excess calcium ions will be transported out. But what will happen when digoxin is present? When we take digoxin, this digoxin will inhibit the sodium potassium ATPase. Now sodium potassium ATPase will be inhibited. it will automatically inhibit this antipotar as well, because if the gradient is not established, the gradient direction changes, there will be lesser sodium ions coming inside, there will be lesser calcium ions going outside. If less amount of calcium ions go outside, there will be reduction in calcium export, and more calcium ions start accumulating in the cytosol, which will lead to enhanced myocardial contractility, because calciums help in contraction of cells, contraction of muscle fibers. So this is the basic functioning of digoxin. Now, there are some other similar inhibitors like obane, which also inhibits the sodium potassium ATPase, and palitoxin, which converts this sodium potassium ATPase into a non-specific cation channel, allow the transport of any cation in a non-specific manner. This was the functioning of digoxin. Now we will see the fundamentals of various transport ATPases. We will briefly talk about these. Let's start with P-type pump. P stands for phosphorylation, okay, and the examples are sodium potassium ATPase, we have calcium ATPase, we have H positive or proton ATPase, and we have various type of cation transporters. V here stands for vesicle, and V-type ATPase is majorly found in vesicles, and it functions as proton transporter. It can be found in lysosomes, endosomes, Golgi, and secretory vesicles. For F-type transport, this is seen, and we all have seen this in electron transport system or electron transport chain, and it is also considered as a reversible coupling device. There can be ATP hydrolysis and there can be ATP synthesis as well. It can be seen in inner mitochondrial membrane, it can be seen in thylakoid in plant chloroplasts, it can be seen in plasma membrane of bacteria. For ABC transporters, ABC stands for ATP binding cassette, and they are mostly carriers, but some exceptions are there where it can function as channel as well. Now let's have a look at these ABC transporters in details. First we will obviously start with the structure of these ABC transporters. This is the generalized structures, and this structure will hold true for most of the ABC transporters. We have two transmembrane domains. Each domain is 6-pass transmembrane receptor. So for total we have a monomeric polypeptide of 12-pass alpha helix. It is 12-pass alpha helix. Even pass means both N and C terminus have to be on the same site, and in this case both N terminus as well as C terminus are on the cytosolic phase. Okay, between the 6th and 7th helix we have a loop where ATP binding domain is present, and this ATP binding domain is commonly called as nucleotide binding domain. Okay, adjacent to nucleotide binding domain we have a regulatory domain, and on the last alpha helix near the C terminus we have another nucleotide binding domain. What is the significance of these nucleotide binding domains? We will see in a while. What is the functioning mechanism of this carrier protein? This carrier protein will be open only when both the nucleotide binding domains are bound with ATP and regulatory domain has been phosphorylated, and the gate will close when either of these two will be ATP hydrolyzed, mean ADP will be bound or AMP will be bound, and regulatory domain has to be dephosphorylated for closing of the gate. This is the working principle, and this is the significance of these nucleotide binding domains and this regulatory domain. Now these ABC transporters can be classified into two basic types. First is full transporter and second is half transporter. The full transporter, two transmembrane domains and single monomeric polypeptide, whereas in case of half polypeptide we have two separate chains, the transmembrane domain 1 and transmembrane domain 2. These are different. We have N terminus here, C terminus here, we have N terminus here, C terminus here. Okay, this is the main difference. The full transporter is seen in eukaryotes and it acts mostly as exporter, whereas half transport is seen in prokaryotes and it can act as importer as well as exporter. We will see some basic details of ABC B-bun, or this is also known as MDR bun, multi-drug resistance transporter, efflux transporter. Okay, so this MDR bun protein is 170 kilodaltons, consists of around 1300 amino acids. It is P-glycoprotein, means phosphorylation dependent. 12 alpha helix domains are there. Exports the hydrophobic drugs, toxins, genobiotics, various antibiotics, anti-cancer drugs, immunosuppressants, HIV protease inhibitors. So it helps in developing the drug resistance, and that is why it is called multi-drug resistance efflux transporter. What is the mechanism of action of these MDR bun proteins? So first of all, what happens is substrate binds to central cavity because it is 12-pass transmembrane protein. ATP binds to nucleotide binding domain and nucleotide binding domain 2. Now keep in mind that structure is similar what we saw earlier as the generalized structure of ABC transport. So first of all, we have substrate binding. Second, we have ATP binding. Third, it leads to conformational change and substrate will be released outside. Protein flips along and resets its conformation. The substrate, what is the substrate? Substrate is all these drugs. We are coming to the exception we talked earlier, the exception of channels, the CFTR. It stands for cystic fibrosis transmembrane conductance regulator. CFTR gene is present on Q arm of chromosome number 7. It belongs to ABCC7 protein family. It is ATP gated ion channel responsible for passive transport. Now this is ion channel. It will transport passively, but ATP will be required for the opening of the gate of this channel. What is the mechanism of action? So protein kinase A phosphorylates the regulatory domain. Right domain 1 and 2 will bind to ATP, which will lead to conformation change, which will lead to opening of this channel and efflux of chloride ions. And this efflux of chloride ions will drive water through osmosis. The ATP will hydrolyze and the channel will reset itself. Now, what are the physiological roles of CFTR? We will study CFTR itself in details in respiratory physiology as well. For the cell biology portion, we will be studying briefly about these roles. It helps in salt and water secretion across the epithelia. It helps in maintaining airway, surface liquid and mucus hydration because it is found in the tissues in which secretions are there, like lung tissue, intestinal tissue, pancreas. There may be sweat glands as well. In case of lungs, chlorine secretion to airway of lumen, which maintains the mucus hydration. In case of pancreas, it regulates the enzyme secretion and bicarbonate transport mechanism. In case of sweat glands, the reabsorption of chloride and sodium takes place. In intestines, fluid and electrolyte secretion is controlled. Now we will talk about a very important mutation, which is delta F508, which is deletion of phenylalanine on position number 508. It leads to impaired transport of chloride and water. This will lead to hydrated mucus. It will lead to concurrent lung infections, and in case of pancreas, it may lead to pancreatic insufficiency. That was about CFTR.